Hello, everyone, and welcome to our second half of the lymphatic system. So on this portion of our discussion of the lymphatic system, not the ecosystem that you may have heard being discussed just a second there, we're going to talk about exactly how this cellular mediated immunity happens. So on the T cell side of things, we talked about how we have antigen presenting cells that can present an antigen on a class 2 MHC marker. If it presents it on a class 2 MHC marker with this cell, this antigen presenting cell is saying kill this, is what's invading us. And if it's on an MHC class 1, then it's a sick cell displaying this antigen on its marker and it's saying kill me. So big things that we learned or talked about, in addition to a whole discussion on innate or non-specific defenses, that we talked about this idea of antigen presentation for T cells. What we're going to do today, or for this session, is that we're going to discuss the different kinds of T cells at length, and then we're going to talk about B cells as well. We'll talk about antibody-mediated immunity. So important aspects of differentiating one class of T cells for another are these clusters of differentiation, or CD markers. There are different types of CD markers found on different types of T cells. First class or type of CD markers that we'll discuss are CD8 markers, and I'm putting a sun here because it's important again. They're found on cytotoxic T cells as well as suppressor T cells. These CD8 markers are going to respond to antigens that are on class 1 MHC proteins. And remember, if it's an antigen that's on an MHC class 1 marker, that says, kill me, I am a sick cell. So if we think about it, cytotoxic T cells, their job is to target cells that aren't displaying their markers correctly. It kind of makes sense that cytotoxic T cells would target a cell with an antigen and an MHC1 marker. So what these markers do, these CD8 markers, CD4 markers, is that they're not only used for identification, but they also allow for communication between cells. So it's these CD8 markers that can recognize an abnormal MHC class 1 protein. CD4 markers are found on helper T cells. They respond to antigens on class 2 MHC markers. So remember, an antigen on a class 2 MHC protein says kill this. So these helper T cells have a role in what's called co-stimulation, and they help to activate other immune cells to make copies of themselves in order to fight this specific infection. So the CD4 markers are found on helper T cells. Another interesting note about CD4 markers, and you've probably heard of CD4 helper T cells, is in a clinical discussion of HIV, and that the HIV virus is able to get into these helper T cells because it matches this CD4 marker, and it can dock onto the cell itself and insert its virus into the cell. Not all of your cells are going to have the docking mechanism, if you will, for the virus to get inside of it, but these CD4 helper T cells do have that docking mechanism. There is a theory, and keep in mind it's just a theory, that one of the reasons there are some people that will have HIV infections and they may have a longer, more productive, better quality of life, if you will. They don't have as many sicknesses if they have the HIV infection as other people do, and it may relate to the fact that their CD4 markers may be slightly abnormal. So maybe the markers that help the virus to dock onto the cell are slightly changed or just not quite the same, so they're not able to affect all of their helper T cells. Both CD8 and CD4 markers can bind to CD3 or cluster differentiation receptor complexes, and that will allow those cells to be activated. But the ones I'm really concerned, CD8, CD4, knowing helper T cells, CD4, cytotoxic suppressor T cells, CD8. So co-stimulation is a process by which helper T cells have to be activated before they can make copies of themselves and do their job. And what this co-stimulation process entails is that even though the T cell may recognize that, okay, that's the antigen that we need to fight, we need to have something confirm that. So the confirmation process is what we call co-stimulation. So for a T cell to be activated, it has to be co-stimulated. So another cell has to release a chemical messenger to stimulate that cell. And it does that by binding at a second site. And usually it's that helper T cell that will help or assist in this process of co-stimulation, which confirms and says, yep, you're absolutely right. You are specifically designed to deal with this specific pathogen. You may activate yourself. So to activate those CD8 T cells, remember those cytotoxic and those suppressor T cells on there, it's activated by the exposure of the antigen on an MHC protein. One is going to respond very quickly, and that produces the cytotoxic T cells as well as the memory T cells. And the other is going to respond more slowly. So remember, suppressor T cells also had CD8 markers, but when they're activated, it takes them a little bit longer time for them to be fully activated, even after the process of co-stimulation, because we really don't want suppressor T cells to be in large numbers while we're actively fighting this infection. So we want a chance for the cytotoxic T cells to do their job and to build up the memory cells so we can have resistance against this pathogen, the same pathogen if we see it again. So we really don't want suppressor T cells culling down those numbers of cells and killing those cells. So the suppressor T cells are going to respond to the activation process a little bit slower, which is what we want to happen, so that we can actively fight the infection. And then after we've actively fought the infection, then we can go about the business of culling back some of those numbers of cytotoxic T cells and just bringing down that load of cells to its normal number. So speaking of cytotoxic T cells, their job is to actively seek out and immediately destroy cells that have an antigen on their class 1 MHC marker. 3 ways that they can do that. They can poke holes in the plasma membrane of the cell, either the bacteria cell or the infected cell, the bacterial infected cell or the virus infected cell, or they can secrete poisons that will destroy the target cell from the outside in, or they can program that cell for apoptosis, so they can activate genes in the target cell to undergo programmed cell death. So there are 3 different ways these cytotoxic T cells can destroy their targeted cell. Their targeted cell is a cell that has an antigen on its class 1 MHC marker. And here's a beautiful picture of this process happening. So we have the antigen recognition, and notice that we have our class 1 marker and the antigen that's on that says the cell is infected. We have to go through this process of co-stimulation so that we have another cell comes along and it says, okay, do you actually recognize this particular antigen? Are you chemically and physically able to deal with this antigen? Once that is confirmed, it says confirms the first signal, then this T cell will be activated, and after it's activated, then the cell divides and produces more cytotoxic T cells as well as memory cells. We want to save some for the next time you see this infection, and then these memory cells will actively target the infection. So then we have the destruction of the cell. Remember, there's 3 ways that we can have that cell destroyed. We can poke holes in it, destroy the plasma membrane. We can say, program cell death. So basically this cell is sick, and when it presents the antigen on its class 1 marker, it says, hey, I'm sick, somebody help me. This is what's making me sick. Kill me, put me out of my misery. So the cytotoxic T cell says, well, which way would you like to go down? I can poke holes in you and cause osmotic lysis to take place. I can program you for programmed cell death, apoptosis, or I can just release a toxin that is going to cause you to starve yourself to death because it disrupts your cellular metabolism. So then you have your cell that's infected that says, you know what, I'd rather go for apoptosis. I've been dreaming about putting myself down for a while. And so then your cytotoxic T cell tells it, you know what, if that's the way you want to go, then go ahead and Godspeed. You've been of great service to the body. And the cell says, thank you, it's a pleasure serving you, and boom, the affected cell dies. Well, I'm sure they don't have that full conversation, but it's something along those lines chemically. So memory T cells are produced with cytotoxic T cells, as we saw in the previous slide. They stay in circulation because the next time we see that same antigen, we want to immediately become cytotoxic T cells so we can start getting rid of this infection. Suppressor T cells secrete suppression factors. Big job of suppressor T cells, as the name suggests, is that they're going to inhibit or suppress the response of T cells and B cells. We don't find in a healthy body suppressor T cells doing anything until we after we have our initial immune response. We want the T cells and B cells to actively deal with this infection, actively deal with this pathogen, and then and only then do we start to repress the T cells and the B cells. So their job is to kind of limit this reaction to just a single stimulus so it's not widespread and we don't have the body, these T cells and B cells, attacking your own tissue. So the activation of these helper T cells, these CD4 T cells, we activate helper T cells. When they become activated, they secrete cytokines or messengers or chemical messengers to help with co-stimulation and also to for them to help activate B cells. We have memory helper cells that are kind of like memory cytotoxic T cells. They just kind of lay in wait for the next time we see this infection. They're just hanging out in reserve. So here, what we're looking at in this picture here is we have this antigen recognition and activation of helper T cells. So we have our helper T cells that are activated, and notice how these active helper T cells are secreting these chemical messengers. And these chemical messengers, or cytokines, can stimulate not only T cells for cellular-mediated immunity, but they can also stimulate B cells for antibody-mediated immunity. We haven't talked about B cells yet. We've only talked about T cells. But understand these helper T cells, we have different classes of helper T cells, can help to activate and stimulate both other T cells as well as B cells. Memory T cells, they just kind of stay inactive, and notice that if it's an inactive helper T cell, it's not releasing these chemical messengers. So that's an important point to make note. So the functions of these cytokines, and we've kind of talked about cytokines since our very first session we had over the lymphatic system, that we've just kind of routinely called them chemical messengers, and that's really just a generic category that they fit into. Some of the jobs of these chemical messengers as they can stimulate cell division, so they can produce memory helper T cells, accelerate cytotoxic T cell maturation. They can attract and stimulate macrophages, attract and stimulate the activity of cytotoxic T cells, and also promote the activation of B cells. So there are various different jobs, and keep in mind that this list is not exhaustive. This is not the only thing that cytokines can do. But in our discussion, from what we're discussing here, it's kind of an what we'll do is kind of an introductory anatomy and physiology, if you will. These are the four roles that we want to target in on as the jobs of cytokines. So now we get into B cell land, and the remainder of this lecture will be over antibody-mediated immunity. B cells come from the same lineage of cells or the same lymphocyte as T cells. They have a slightly different role. T cells have a role in cellular-mediated immunity. They will directly kind of cell-to-cell combat, if you will, or hand-to-hand combat. They will directly address the cells that are infected. With B cells, it's more of like the snipers. So if you kind of think of it as a military campaign happening in your body when you're fighting an infection, it's that you have your ground troops that engage in hand-to-hand combat, and then you have your snipers. B cells would be your snipers, and their bullets are going to be the antibodies that they release. So they're going to attack antigens by producing very specific antibodies towards that specific pathogen. There are millions of populations of B cells, and they each have different antibody molecules. Although we only have 5 different classes of antibodies, there's a lot of variation within each class. So there are tons of different combinations and different types of antibodies that are produced. Preparation process of activating these B cells is what we call syncytation. Having the antigen that the appropriate B cell is sensitive to is what will trigger its activation process. So, speaking of which, we have a beautiful picture to illustrate it. We have our antigens that are circulating. Those things, those foreign objects that can make us sick, they're part of a bacteria or a virus. And we have our class 2 MHC marker on our B cell. Remember we said we find class 2 markers on antigen-presenting cells as well as lymphocytes. A B cell is still a lymphocyte, so we have that marker that's there. And if we have the appropriate antibody that's attracted to or sensitive to this antigen, that antigen will bind to the antibody. And this forms another one, like we saw for the complement in the previous session. An antigen-antibody complex is formed here. After we have that complex that's formed, we can process that antigen, and if we present that antigen on its class 2 marker, we have not only allowed for antigen presentation to happen to activate T cells, but we have also now are allowing for the B cells to be activated as well. So the helper T cells, their job or role with antibody-mediated immunity, as we refer to B cells, is to help the B cells to become activated. So the helper T cell will bind to that class 2 MHC protein that's holding the antigen, releases a chemical messenger that says, yep, it's kind of like co-stimulation. That says, yep, you're right, you are sensitive to that antigen, you have the antibodies to deal with it, go ahead and get activated and divide and make antibodies. So now we have our T cell that recognizes the antigen on this MHC1 marker of our B cell. The T cell, the helper T cell, releases a cytokine or chemical messenger, co-stimulates that B cell, so it gives it the go-ahead and says, yep, you're right, you match this antigen, go ahead and make copies of yourself, and then fight this infection. Godspeed to you. So now when those B cells are ready to activate, not only do they activate, they now are turned on, but they're also going to divide or differentiate into two camps. We have plasma cells and memory B cells. The plasma cells are going to actively make antibodies to deal with this active infection that we're encountering, and the memory cells are going to just kind of lay in wait and be inactive so that the next time we see this antigen, we're ready to go. And here's that differentiation process happening. We have our division and differentiation where we have our inactive memory cells. Then we have our activated B cells that become plasma cells, and it's these plasma cells that release these antibodies or produce the antibodies. And remember, these antibodies aren't just made willy-nilly. They're made in response to the specific infection that a B cell was sensitive to and was stimulated by the helper T cell to make more copies of itself. Great short answer question, once again, would be to explain the process of T cell activation, B cell activation, how does antigen presentation work, and how does sensitization and activation of B cells work. So those are excellent short answer questions. So those antibodies that we talked about are specialized proteins, and every antibody has about the same basic structure to it. There are two parallel chains of polypeptides, and they're usually in this Y configuration. Now, this used to all be theorized, but now we know it to actually, by the advances that we've had in technology, we can see the structure of the proteins, and it does, in fact, make a really cool little Y shape to it. You are left with the heavy chains, which are the longer of those parallel lines or parallel polypeptide chains, and then you have the light chains, and they're the shorter ones. At the end of each of those chains, you're going to have a variable region, and you're going to have a constant region. The constant region is going to stay the same for every antibody in that class, and there are 5 classes of antibodies. The variable region, on the other hand, is going to differentiate or be different for each individual antibody, and that's where the specificity comes in at the variable region. Well, that's where it really comes into play at the variable region. So the 5 different classes of antibodies that have the same, like all of the antibodies in one particular class have the same heavy chain that or constant region, are IgG, IgE, IgD, IgM, and IgA. You will have to tell me be able to list all 5 of these antibodies, and there are some things that you should know about them that we're going to discuss in a second. What hapten molecules are is that they're actually just partial antigens, and for a hapten molecule to become or act as a complete antigen, it has to attach to a carrier molecule. And sometimes that carrier molecule could be even something as simple as penicillin, or it could be latex. It's just anything else that this hapten molecule can attach to, and it acts as an antigen. So if it's acting as an antigen, your body doesn't recognize it, so it's going to elicit an immune response. So your antibodies will attack not only the hapten, but it'll also attack the carrier molecule. And let's say that the carrier molecule, that penicillin just happens to bind to a normal cell, it will attack that normal cell, and you'll have it'll elicit an immune response as a result of that. So dangers of haptens is that it can, people that are, like, they say they're allergic to penicillin, this is why they're allergic to penicillin, or they're allergic to latex, is because any portion of that penicillin or latex can attach to their normal cells and cause their normal cells to appear to be antigens or pathogenic, and it will trigger an antibody response so that the antibody attacks the normal cells. So for our first class of antibodies, we're going to talk about IgG. Bring the old sunball out here, things that are important that you remember. It is the largest and most diverse class of antibodies, so it has the most variability within this class. Approximately 80% of all antibodies are in this IgG. IgG is responsible for resistance against lots of different viruses, bacteria, and toxins. Also very important, so far there are 3 bullets out of this slide that are super, super important that you should remember, is that IgG can cross the placenta, and it provides this passive immunity to the fetus during embryological development. And also the anti-Rh antibodies created by Rh-negative moms. So remember, if you're A negative, and you're pregnant, you have to get that Rogam shot because you don't want your body to make antibodies against your child. The anti-Rh antibodies that if you're negative that you would make, they're IgG antibodies, and they can also cross the placenta, and that can cause hemolytic disease to the newborn. If you have a positive blood type, you don't have to worry about that. But it's only if you have a negative blood type and you're female carrying a child do they give you that Rogam shot to keep these IgG antibodies, and let's start it, we've talked about this before, from crossing the placenta and attacking your fetus. Next class, IgE, they attach as an individual molecule, and they can be attached to the surfaces of basophils and mast cells. When an IgE antibody is bound, it causes the cell to release histamine and triggers an immune response. So IgE is very important in allergic responses. So just these 2 items here are really super important for IgE. For IgD, it's an individual molecule on the surface of B cells. It can bind to antigens in extracellular fluid, and the binding of those antigens can play a role in the sensitization of those B cells. So IgD is kind of the least super specialized of the classes of antibodies. For IgM, although it is not the largest class of antibody, nor is it the most diverse like IgG, but it is the first class of antibody that's secreted after an antigen is encountered. So once we see this bacteria, this virus, this thing that ain't supposed to be there, IgM is the first class of antibodies that's secreted. Now, after IgM concentrations start to decline, well, as they're declining, IgE production is accelerating there. So IgM kind of first on the scene, IgG is going to be the next one on the scene. So its kind of claim to fame is that it's the first one secreted after we encounter an antigen. Plasma cells secrete individual IgM molecules, and it acts as a 5 antibody starburst. And that is more of a structural discussion on what this antibody looks like. Anti-A and anti-B antibodies, so that if you have type A blood, you have anti-B antibodies. If you have type O blood, you have anti-A and anti-B, and the whole blood typing business we talked about. they are responsible. They're actually in this class of IgM antibodies, so that's also important. And IgM antibodies may also attack bacteria that just might not be sensitive to IgG. So another good reason for having these guys be first on the scene, because there may be some bacteria that aren't so sensitive to IgG. We're the largest and most diverse class of antibodies. And our final one, IgA, is primarily found in glandular secretions such as your mucus, your tears, saliva, semen, vaginal fluid, all that good stuff. They will attack pathogens before they gain access to the interstitial tissue. That's why we find them in your not plasma membrane, but your mucous membranes, places that you're going to secrete stuff out of to kind of keep things out of the box. They can also circulate in blood as individual molecules, or sometimes you find them in dimers or in pairs. So your primary and secondary response to antigen exposure, kind of keep this in mind that, first time, shame on you, second time, shame on me. So there are these memory cells that are produced. So not only do we have a primary and a secondary response for antibody-mediated immunity with the antibodies that we just talked about in the B cells, we also see it for cellular-mediated immunity. And that we understand because we saw memory T cells and memory B cells. The first time that you're exposed to this antigen, this bacteria, this virus, this pathogen, this thing that's making you sick, is what we call your primary response, how your body primarily responds to this infection. The next time that you see it, it's going to trigger a secondary response. Secondary responses are extensive and they're much more prolonged. Reason being, you already have memory cells that are primed and ready to go for this, so that the way that and we're not saying the infection is longer, we're saying that your immune response is longer. And sometimes your immune response is so robust that you don't actually even feel sick, if you will. You might have a sore throat for a day, or you might not have a sore throat at all, because your body has responded to this antigen so quickly because you have these memory cells. So in the primary response, the peak of that response can take 2 weeks to actually develop. Remember, we got to round all these cells and activate them and co-stimulate them, and we've got to recognize this antigen and figure out who's supposed to do what and all this good business. So to get the peak response there where you start to maybe feel a little bit better, or even slightly, you're starting to feel better, but before you're actually on the 100% mend, it can take some time to develop. And then it's going to decline very rapidly. The IgM antibody is going to be first on the scene, as we talked about in our discussion of the different classes of antibodies, and it'll be produced faster than IgG. Although it's not nearly as effective as IgG, it is a good first responder, so it will be first on the scene there. Now, in your secondary response, notice the difference we have here. So the second time you've seen it, first time you saw it, IgM was first on the scene. Second time that you see it, for your secondary response, we have IgG is first on the scene there, because we have these memory plasma B cells that are able to activate very quickly and make these IgG antibodies, which are much more effective. So we are able to see for the secondary response that not only does IgG now become the first antibody that's on the scene to deal with the infection, which is different than we saw from the primary response, but we also see that you have a much longer exposure of the antibody to the antigen for a longer period of time. So that's what we mean by it's a prolonged, more extensive exposure or immune response. And just to remind you all and show you what I mean by the discussion we had in the very first session, that just because we're talking about innate immunity and adaptive immunity, or specific and nonspecific immunity separately by themselves, does not mean that they happen in a vacuum. This picture is a great picture to show how your body responds to a typical bacterial infection, where we have immune cells that are part of your nonspecific defense, neutrophils, and the specifics again, cytotoxic T cells, all kind of mounting together so that by the time you're a week into this infection, you start to see kind of your peak immunological response to it. And then as you have all of these other cells, neutrophils acting as phagocytes, natural killer cells that are killing any abnormal virus-infected cells, well, this is a bacterial infection, but that are able to kill any cells that are harboring the bacteria inside of them, they're able to do that. Macrophages that can act as antigen-presenting cells, cytotoxic T cells that can target these cells, and these MHC1 markers, plasma cells or B cells that are made, and they can make their antibodies. And then we have the antibody titer go up even higher. So this is just a great picture or illustration of how all of these things that we've talked about, the innate and the adaptive defenses working together simultaneously to elicit an immune response. And these are the learning outcomes for your tests that will happen in a couple of weeks here. I'm not going to go over those, but make sure that you study them and kind of make that part of your preparation for your exam. Next chapter that we'll go over is Chapter 23, Respiratory System. So I will see you all for our next session. Have a great day.